thanks for coming. So, like Dirk said, I'm going to be talking about mapping, taxonomic, and thematic naming errors post-stroke. Just a brief introduction on my background. So I did my PhD with Dr. Ludwig Desai, where I had the opportunity to work with fMRI and brain stimulation in neurotypical adults, looking at concept representation in the brain. And I also had the opportunity during that time to do a lot of work with C-STAR using lesion-symptom mapping techniques in people with post-stroke aphasia and looking at how damage and disconnection affects language. And then now I'm currently in my postdoc here at University of South Carolina with the aging brain cohort and still doing a little bit of work with C-STAR. So here's an overview for the talk. I'm going to talk about the background and the motivation behind this work, a literature review, and then talk about the experiment itself and the results of the discussion. And so this is work that I'm currently working on. It's not published yet, so I'm very happy to hear about any thoughts that you might have about it. So why is it important to study naming impairments? First of all, it's extremely common in post-stroke aphasia. Regardless of the type of aphasia, it is one of the most common impairments that people report having. It's extremely important for quality of life and psychosocial well-being in people with post-stroke aphasia. So word finding difficulties is a good proxy for overall language impairment. So people that have naming difficulties tend to also have poor communication, and it affects their quality of life. It's the main focus of a lot of therapy protocols, and naming is a good way to measure treatment efficacy. It's replicable, it's relatively easy to administer. You can say they named 5 more objects than last time, and it's a good way to measure progress, I guess. So all that considered, it's important to understand the neurobiological basis of naming difficulties. So some error types broadly defined now. What are we talking about when we're talking about naming errors? So these are 3 main types. There are subtypes to each of these. So there's phonological errors, where they delete or insert phonemes of the target word. So for example, they see ghost, but they say goth. Then we also have semantic errors, which is a real word noun that is related. It doesn't have to be a noun, it can be any type of word, but it's related to the target. So instead of banana, they say apple. So it's related because it's a fruit. And then we have mixed errors. So this is when a real word is related to the target both semantically and phonologically. So they see a cat and they say rat. So they substitute a phoneme and they also say an animal. And this project, this work today, is going to be focusing on the semantic aspects of these errors. So how can we model speech production and why these errors might occur? So a brief background is one of the first models was Levelt's lemma model. So this was concrete stages from conceptual preparation all the way to naming and word production. So each of these steps is its own feed-forward process, and these processes don't interact with each other as you go down this pipeline from conceptual preparation to speech. But the issue with that model is that it has some difficulty explaining mixed errors that have both a semantic and a phonological component. So then the Dell's interactive 2-step model was proposed. So this allows interaction between semantic features and phonological features to create both semantic errors, phonological errors, and those mixed errors. And then there's been work done on what are the neural correlates of these different levels in the speech production model. So the semantic features have been mapped in previous work to structures such as the anterior temporal lobe, the angular gyrus, and the frontal cortex, whereas the phonological layer has been mapped to sensory motor cortices and the supramarginal gyrus, among other areas. And so this is going to be an ongoing theme, I guess, today in today's talk, is the dual stream model of speech, where the ventral stream is related more to semantics, and the dorsal stream is related more to phonological steps. And so if we want to map these things onto the brain, we can look at some models of the neurobiology of speech. So this is the Hickok and Popple model. And so we can see here, again, this ventral stream is where we might expect disruption of those ventral areas might be related more to semantic errors in naming. And we can also look at a model that might be more specific to confrontation naming. So here you can see a little puppy down there in the occipital lobe. So that's a picture that they might be seeing, or an actual puppy. And then it gets fed forward into the ventral stream, where the lemmas of the conceptual processing is done. And then it's fed into the phonological and syllabic, and making the syllables to say the word. And so these different areas can often interact with each other. It's not necessarily a straightforward process from one to the other, but these are the kind of regions of interest that we might be interested in looking at in naming. All right, so now back to my specific project, which is going to be about semantic errors, like I said before. So within semantic errors, we can break that into two different types of sub-errors. So we can have thematic errors or taxonomic. So the thematic or the associative errors is when the word that the participant says is related to the target word via events or actions or other kind of associations, right? So they see a picture of a dog, but they say leash because we put a leash on a dog and we walk it. Or they say bone because dogs chew bones, right? Taxonomic errors, on the other hand, are when they say a word that's related to the target via categorical properties. And a lot of these tend to be related to shared perceptual features, but not always. So there's different levels of taxonomic errors. For the purpose of this talk, they're all going to be grouped into taxonomic errors. We're not going to talk about individual, subordinate, or coordinate. So they see dog and they say animal, for example, or dog and they say cat. So now let's get into the literature review of what work has been done already in this space. So a very seminal and important study was the Schwartz et al. 2011. That was published in PNAS. It was a voxel-based lesion-symptom mapping study in post-stroke aphasia, and probably, I believe, the first in post-stroke aphasia to look at this thematic versus taxonomic distinction. So they basically found that when you have these patients that are making taxonomic or thematic errors and you regress those error types out from each other, meaning you separate them statistically, damage in the anterior temporal lobe was associated with more taxonomic errors, whereas damage in the angular gyrus / temporoparietal junction area was associated with more thematic errors. And so one important aspect of this study that I want to point out right now, because I'll talk about it a little bit moving forward, is that they also controlled for pyramids and palm trees performance. So they did this to focus on the word retrieval aspects of naming, while kind of accounting for patients who might have worse nonverbal semantic capabilities. And so we can also look at evidence from semantic dementia. So for those of us who are unfamiliar with semantic dementia, it's associated with deterioration of the anterior temporal lobes bilaterally, but as the disease progresses, it also leads to widespread atrophy elsewhere, but it does tend to be more focused in the anterior temporal lobes. And these patients tend to make increasing amounts of taxonomic errors with a relative preservation of thematic knowledge and thematic associations. So this has led to kind of a hypothesis that taxonomic knowledge is housed or stored in the anterior temporal lobes. And so these patients, as the ATLs atrophy, they then tend to rely on the thematic associations that are preserved elsewhere in the brain. And so here you can see just some sample images from patients with semantic dementia, and you can see that atrophy in the bilateral anterior temporal lobes. All right, so now fast forwarding a little bit to a very, very good and comprehensive review, in my opinion, of taxonomic and thematic knowledge in the brain, Neerman et al. So this is only a snippet of the review. They also review developmental studies and all sorts of other evidence from other areas of study like linguistics. But in their review of the brain imaging part of it, they concluded that after the review of 28 of these brain imaging studies, a promising but preliminary hypothesis is what they call a dual hub hypothesis. So dual hub meaning that one hub of knowledge is the anterior temporal lobe, where that area specializes in taxonomic knowledge representation, and then the TPJ, or temporoparietal junction, is extremely important for the thematic associations. And here in this image you can see taxonomic is connected to color and shape properties of words possibly, which might be the driving factors between taxonomic categorization of things, and then the thematic associations, on the other hand, might be more related to action or spatial location, for example. All right, so now, since the 2017 review that concluded this was a promising hypothesis that needed more testing. So there's been more testing, and it has not really cleared up the picture very much. So this is from a meta-analysis of neuroimaging studies that was just from last year, and they found evidence against the dual hub hypothesis. So this red arrow here is pointing towards a cluster in the anterior temporal lobe that is reliably activated for thematic greater than taxonomic conditions in these neuroimaging studies. So that is directly against what the dual hub hypothesis would predict, right? And even when they lowered the statistical threshold to a really liberal value to see just if there's anything in the anterior temporal lobe for taxonomic conditions, they didn't find it. So very, very weak evidence for taxonomic processes going on in the ATL. And the only cluster that they did find for taxonomic specifically was in the right lingual gyrus. So not really an area that had been hypothesized, but that's what they found. And then more work actually this is from our group. Blackett et al. Used connectome-based lesion-symptom mapping. So that's basically looking at white matter connectivity between brain areas. And they looked at confrontation naming, errors in post-stroke aphasia. And they found that weaker connectivity between ATL and some other areas like the posterior cingulate and the insula was associated with more thematic errors. So this on the left here is the MTG pole to the precuneus, or posterior cingulate, sorry. And then on the right is the inferior temporal gyrus to the insula. And we can see here on the x-axis, weaker connectivity is 0, stronger is 6, and as it approaches 0, thematic error proportion increases. So again, evidence that is pretty directly against the hypothesis that the anterior temporal lobe is specific for taxonomic knowledge. So, in a nutshell, overall summary, there's divergent results. The dual hub hypothesis is not really being perhaps supported as much as we might have thought back in 2011. And the status of what's going on with these different knowledge types in semantic errors and semantic processing is kind of unclear. So how do we move forward from here? Well, using multiple and complementary methods might help. So instead of just using voxel-based lesion symptom mapping. Maybe we can use some other methods in post-stroke aphasia. Related to that, a lot of prior work in post-stroke aphasia specific to taxonomic and thematic errors is that it has been limited to the left hemisphere, which makes sense because a lot of patients are unilateral left hemisphere stroke. But now, as our imaging procedures have improved, we have ways to actually tap into the health of the right hemisphere as well. So maybe looking at the right hemisphere will reveal something that we haven't seen. And like I mentioned before with the pyramids and palm trees test, the previous post-stroke aphasia studies have included that as a covariate. So the question then becomes, is that necessarily desirable if we're looking at semantic naming errors? Does it make sense to kind of regress out and get rid of variability between patients in nonverbal semantic abilities? Maybe that's not desirable, because maybe those nonverbal semantic abilities are actually informing the naming process, right? So in this work that I'm about to show you, I did not control for pyramids and palm trees, and instead I just regressed taxonomic from thematic directly. So what are the goals of this study? So the first one is to obviously map the neural correlates of taxonomic and thematic confrontation naming errors. And like I said before, I want to use complementary methods. So I'm going to use the straight up voxel-based lesion symptom mapping, which a lot of us are familiar with. But I'm also going to use two newer techniques, or techniques that are at least used less in the literature. So one is an activation-based technique to map errors. So this is going to be actually based on task fMRI in people with post-stroke aphasia. And I'm also going to use a network-based measure, which is related to connectome-based lesion symptom mapping, but is not the same. And I'll get to both the specifics of both of those things more in a bit. And hypotheses. So the first major one that I had going into this was that damage or disconnection of both the ATL and angular gyrus in the left hemisphere would be associated with thematic errors specifically, and not necessarily taxonomic, just based off of the most recent research that has been done. And then the second hypothesis was that maybe we'll find, using the activation-based or the network-based methods, we might find neural correlates in the right hemisphere that are related to taxonomic errors specifically. And this is based on some work done in healthy adults using fMRI that has shown that taxonomic conditions in tasks actually have more activity in the right hemisphere than thematic conditions. So, brief overview of the participants. 79 adults with chronic aphasia. Here you can see the breakdown of the aphasia subtypes and the number of those in our cohort. All of them had left hemisphere unilateral stroke. And my main measure of interest is the Philadelphia naming test. So for those of us who are unfamiliar with that, it's 175 items that the participant must name. And this was administered twice at baseline. So when they first came into the center, and it's administered twice, one on one day and one on a separate day, to account for day-to-day variability in naming performance in people with aphasia. And then those two scores were just average. So the scoring and coding was all done by trained graduate research assistants and certified speech-language pathologists who first coded the error types according to Roche's original guidelines that come with the Philadelphia naming test. And then after they're coded according to phonological or semantically related, etc., we break them into semantic and mixed errors specifically, because those are the error types that have the most semantic component, which is what we're interested in here in this study. And so within those errors with the semantic component, they were broken into taxonomic and thematic error types by trained GRAs who were shown how to do this, and we checked inter-rater reliability and all of those things. One important thing to note is that the way that this is done, we just followed the way that it's been done in the past. It's debatable whether this is desirable or this is the way it should be done, but we just followed previous literature. The errors, if you come across a semantic error, it's first checked if it's taxonomic. So let's say they saw a picture of dog and they said cat. That's going to be categorized as taxonomic because it is an animal. But that also has a thematic component, right? Because they're pets. You encounter cats and dogs in similar situations. They're related in a lot of different ways, but this is just the way taxonomic and thematic are usually coded. So if they saw dog, said cat, that is considered a taxonomic error in this study. And then from that, we created error proportions. And again, it's debatable whether this is the best way to measure error types in picture naming. So some people prefer to just do a total count of error types, but here we made a proportion of taxonomic or thematic errors out of the total errors that that participant made. So this comes out with tax prop and theme prop is what I'm going to call it moving forward. And so for all the neuroimaging analysis that I'm going to talk about, these two error types were regressed out from each other in addition to lesion volume. And we can see here the distribution of taxonomic and thematic errors in our cohort. So this is very typical. Taxonomic errors are more common than thematic errors in both healthy people and usually in people with post-stroke aphasia. There are ways to experimentally elicit more thematic errors if you use distractor items, different priming paradigms, but we didn't have those things here, so this is the distribution of error types. Then the symptom mapping methods that I used. So each of these have pros and cons, or things that are good and then maybe limitations that you could say about each of them. So the pros are in green, and then below those are the limitations of each method. So voxel-based is straightforward. It maps necrosis with behavior, so damage with a behavioral outcome. So that's good, but you're limited to areas only within the stroke territory. So you can't really look at undamaged areas, right? Because there's not damage in the undamaged areas. The activation-based method, which is again based on task fMRI, is good because it maps how residual brain function during a task actually relates to performance in a task that is done outside the scanner. But the negative, as we'll see when we get to my results of discussion, is it can be a little bit difficult to interpret. It's not as straightforward as damage here equals bad performance. And then the network-based method, which is, again, using white matter connectivity. The good thing is that it detects effects related to disconnectivity of spared regions, so maybe in the right hemisphere, or even spared regions in the left hemisphere. But the negative is that there are tens of thousands of connections to consider, so the correction for multiple comparisons can be very harsh. So I'm going to spend less time on the VLSM because most of us are familiar with it. But basically, a t-test is conducted at each voxel, comparing undamaged performance to damaged performance. Again, we use the residuals for all of these, controlling for lesion volume, and everything moving forward is permutation corrected. And here we can see an image of the lesion incidence map, and this is very typical for studies in the post-stroke aphasia. So the red here indicates the largest number of patients that had damage in a given voxel. So in these red areas, close to 60 people have damage to those voxels. Then as we move out, less and less people have damage. All right, so getting to the activation-based method that I'm using here. This is kind of a breakdown of how this works. So we do fMRI collection during attempted naming. So they do a naming task. Importantly, this task is not the Philadelphia naming test. It's using completely different stimuli, so it's an independent task. And I say attempted naming because while we do have the actual recorded speech of what they said in the scanner, for the purpose of this, we didn't go through correct or incorrect or what type of error they made at a given object. We just take the average of the bold signal that was collected during their attempted naming. And so this is all pre-processed in our pipeline. The lesion components are extracted, and we get a mean fMRI image created for each participant while they did this naming task. Then we, outside the scanner, administered PNT separately. And then we basically do lesion-symptom mapping, but using the mean fMRI image as the input. So a general linear model at each voxel relating to tax prop or theme prop, and then permutation corrected, and then we get the clusters where bold during this naming task relates to behavior. And so this is just some extra details. So the data collection was done using a sparse sampling technique to get around motion artifacts and the problems that come with collecting fMRI data during an actual speaking task. Again, we removed the lesion artifacts, and then we limited the analysis to a bilateral mask, which you can see here. So it includes the inferior frontal cortex, most of the temporal lobe up into TPJ regions. And what you can't really see is we also included some medial parietal regions like the precuneus, and some medial temporal regions like the cortex. All right, and now I've covered VLSM, I've covered the activation method. Now let's talk about the network-based method. So it's a close cousin to connectome-based lesion-symptom mapping, which looks at how white matter connections relates to behavior. But ours differs just a little bit. So we parcellated the brains according to the Johns Hopkins University Atlas. Then we use probabilistic tractography to determine connection strength between the parcels. And this results in, for each participant, a 110 by 110 connectivity matrix, where the cells represent connection strength between any given two regions, two gray matter regions. And then with that connectivity matrix, now is where it starts to differ from traditional CLSM. We're going to use something called the network-based statistic to analyze this. So what is this? It's something that was pioneered by Zaleski back in 2010, and it's been continued to be used, but maybe not quite as commonly as connectome-based lesion symptom mapping. It's basically the graph theory equivalent of voxel-based clustering methods. So here we can look at this analogy. So voxels are to clusters as in the network-based statistic, edges are to subnetworks, with edges being these connections between the nodes in your overall network. So statistically, just briefly, the steps of doing this are each edge's association with the behavior is tested individually. So that's exactly like CLSM. But then you set a semi-arbitrary edge level threshold, like you would do in a voxel-wise analysis, where usually you would set it to p is less than 0.001, right? So you set an edge level threshold, and then the largest contiguous subnetwork of connected components that survive that threshold is identified. And then that's compared to randomly permuted data and what are the largest components identified through randomly permuted behavioral data. And so the advantage of this is that it actually has a power, its ability to detect effects actually increases with the size of the original network. So it's ideal for looking at large-scale whole-brain connectivity networks, whereas CLSM is not as good if you're looking at a whole-brain connectome with tens of thousands of connections. All right, now the results. So the vanilla VLSM analysis in panel A are our results, and that is a cluster where damage was associated with a higher proportion of thematic errors controlling for the taxonomic errors in the lesion volume. And what I have in panels B and C are clusters identified in recent neuroimaging studies in healthy adults that where thematic relations or thematic conditions have greater activation than taxonomic conditions. And we can see that there is a pretty striking overlap between what we found and the clusters identified in these fMRI studies. And this cluster does fall, depending on how you define the anterior temporal lobe, half of this cluster is within the anterior third of the temporal lobe, which is a common cutoff used to say where does the anterior temporal lobe start. So now the activation-based results. Sorry, let me go back one slide. Just wanted to note that we didn't find any clusters that related to tax prop compared to theme prop. So we only found it for theme prop. So activation-based actually was the only method that we used that revealed associates of taxonomic proportion errors. And so this is where lower bold signal during that naming task actually relates to greater proportion of taxonomic errors. So these areas are less active, and that's associated with making more taxonomic errors. And so clusters are found in the bilateral precuneus, bilateral angular gyrus, and the right IFG pars triangularis. And this was surprising because if you recall from earlier, we would expect angular gyrus to be more related to thematic error proportion. But here, using this method, it was more related to taxonomic. And now the network base. So there are a lot of connections here. Yes?
I mean, I guess it's an interesting question because you're saying these are rodents where more activity means better performance. I mean, I'm still trying to work this through. This is fMRI. These are people, some of these people have injury in part of this, right?
So the lesion artifacts have been removed, but yes, like some people do have damage. For example, to the left angular gyrus. Right hemisphere is not as much. Yeah.
And also the medial. Yeah.
Yeah. And then the network-based method revealed a subnetwork, kind of localized, or most of the nodes are in the anterior temporal lobe, connecting to some right hemisphere areas, and then also some areas in the parietal lobe in the left hemisphere. And this is where weaker white matter connectivity is associated with greater thematic error proportion. And so this subnetwork, you might look at it and say, oh, that's a lot of connections. What can we make out of this? Well, we can look at some complex network measures and see what might be, what are the most important nodes within this subnetwork. So we did that using degree and betweenness centrality. So those are measures of basically how central nodes are within a subnetwork. And you can see here that a lot of the most central nodes in the subnetwork, or the nodes that have the most connections kind of going out of them, are areas in the lateral and medial temporal lobe anteriorly. So MTG pole, entorhinal cortex, fusiform gyrus, and the ITG. All right, so now to the discussion. What can we take from this? So damage and disrupted connectivity of the anterior temporal lobe is associated with greater proportion of thematic errors. And this was converging evidence from the VLSM and the network-based statistic. There was a striking overlap with clusters identified in previous fMRI studies in healthy adults. Again, this is some further evidence against the specialization of the ATL for taxonomic knowledge. And surprisingly, we had limited evidence of the angular gyrus being involved in thematic error proportion. And then this also kind of mirrors previous studies of the difficulty of tracking down the neural correlates of taxonomic errors. we did find those results in the activation-based method, but that's like kind of Chris was saying, it's a method that's a little bit trickier to interpret what's going on, and you know, you would kind of want that activation-based method to also be backed up by maybe VLSN or the network-based method as well, to make, if you want to make stronger claims about the neural correlates of taxonomic errors. And it did kind of back up the hypothesis, my second hypothesis, that taxonomic errors might be related to the right hemisphere somehow. Those were some clusters that we found in the right hemisphere that were related to taxonomic error proportion. So partial support there for hypothesis 2. And then the limitations. So interpretation of what a thematic and a taxonomic error actually is tricky. So if somebody sees dog and they say bone, does that mean that there's a degradation of thematic knowledge? Not really. It almost seems that it's a preservation of thematic knowledge. They know that bone is still related to dog, so they say bone instead. But you know, sometimes it might actually be or represent a loss of thematic or taxonomic knowledge. So when interpreting the results, you have to be very careful about how you kind of word these things and how strong you want to make your claims. Yeah, and then the other thing is the question of compensatory strategies. So at what point are these people with aphasia actually using something that was taught to them in therapy, or they're choosing to say a different but related word because they can't find the word that they want to say, so they say something that in real life would be very helpful at communicating, right? Another limitation is this is a retrospective study, so a lot of our measures are used clinically, but maybe not as sensitive as you would want them to be to tease apart thematic and taxonomic errors and what's actually going on behind the scenes cognitively. So in a lot of behavioral studies previously, they use like priming paradigms in healthy adults and look at reaction times, or they'll put distractor items that are thematically or taxonomically related and see how that affects reaction times or accuracy. So ideally, to make stronger claims, we would want a task like that. And then future, what I would like to do is, instead of classifying thematic and taxonomic errors binarily, like it's currently done, we now have the ability to actually make similarity, representational similarity metrics using different things like word2vec or things like that, where we can actually say how similar and in what ways are the word that they said related to the actual target word. So that way we could have a continuous measure maybe of how taxonomically or how thematically related the word that they said was to the target. And that's all I got for you today. So thanks to all the co-authors and the funding. A special shout out to Abby Broadhead, who was actually an undergraduate that I mentored on this project. So she actually did some of the analysis herself. And so that's exciting. And then thanks to all the participants in the SLPs and the GRAs.
Thank you. So, thanks a lot. I think we can go to questions from the room first, and then we'll see what's left online.
Yes.
I had a question about your choice of measures, which I'm sure you'd be interested in. So instead of using the number of errors, you created a proportion. So I just talked to ChatGPT, and it said looking at correlations between brain activity and the number of something versus the ratio of two different things is different than looking at them separately. It can be different and mean something different for a lot of reasons. So are you the first person to use this, and why did you choose that proportion? And I know we have used proportions a lot in our lab.
Yeah.
Like proportions are a little bit something we like to talk about.
Yeah.
Give us your thoughts in a little more detail.
If you don't mind repeating the question.
Okay. Yeah, so Roger asked, We chose to use proportion of errors instead of an absolute count of errors. Were we the first to do this? Has it been done before? What are maybe some pros and cons of doing proportion versus absolute count? And yes, this is a very contentious topic in post-stroke aphasia literature, whether you should use absolute error counts, proportions, what's the best way to measure these things. And a lot of people have very strong opinions, but short answer is no, we're not the first to use it. A lot of people do use the proportion instead of absolute counts. We definitely will want to replicate this with the absolute count, because that would be the ideal finding, is that they're the same in both. Likely won't be the case. But yeah, it's you know, this is the first time I've used proportion error. You know, there's pros and cons, and it's just tricky. So yeah.
Thank you for a great talk, Nick. Really great work. I wonder, and this may be a naive question, but in our sample, you see that participants make more thematic errors, more taxonomic errors, or are it the same pool of participants that's making both error types?
Yeah. So
Again.
Sigfus asked in our cohort, what's the distribution of taxonomic versus thematic errors? Do some people tend to only make thematic or only make taxonomic errors? And I can go, so the distribution of errors, taxonomic is more common. And this is typical of most things except for semantic dementia, where there tends to be a flip to more thematic errors. And there are very, very few, if any, subjects that make proportionally more, we can see here, the very few subjects make proportionally more thematic errors than taxonomic. So, you know, the taxonomic error proportion caps out at 0.2, whereas the thematic caps out at 0.6, right? So, if you're making taxonomic errors, you might make thematic errors, but there are not many people that make only thematic errors.
I had a question. I was glad you pointed out at the end, which you put on a limitation, but I think it's more a point of discussion, right? So what seems to be maybe even a qualitative difference between taxonomic errors and thematic errors, and not exactly this, they may not be that directly comparable, right?
Yeah.
And so I was wondering, which is a topic in lesion-symptom mapping related to the first hypothesis you presented based on the Schwartz et al. Study. So they show damage to anterior to ATL occurs in patients who make more taxonomic errors, right? And then it seems as if the most logical conclusion then is, okay, that area supports taxonomy. But I would argue that's exactly the other way around. That means whatever is impaired through that damage, these patients are now relying on their taxonomic relation. They can't get the actual word, right? And now that they're just relating, they're just giving you everything else they know about the word, which means their taxonomic information is intact rather than damaged. What is damaged is that they're not coming up with the word for dog. So the hypothesis is exactly the opposite, right? And I'm not sure, I think that is different for thematic errors. There you could say if someone makes a thematic error, so they're giving you the wrong word, cat for dog, that means there's something in that area that supports picking the right word.
Yeah. Yeah, it's really fascinating to think about. You're absolutely right that you got to be really careful, because damage and saying, oh, they make more taxonomic errors, it's affecting taxonomic knowledge. It's not that straightforward. And it would be really fascinating to try to tease that apart by, what is an individual person's actual go-to method to compensate if they have a word finding difficulty. Are they more, do some people tend to say bone because that helps them communicate better, whereas another person might resort to say dog or animal instead of dog, right? So it might even come down to some top-down processes and selection of strategies. There's a lot going on for sure.
That's a little bit comparable to something else we find, right? Some people, when they can't find the word, they will not say anything rather than make a mistake, right? Whereas other patients are willing to give their best shot.
Yeah. Exactly.
And any others from the room? I don't see any other questions online. Natasha McClendon says great work. And so if there's no other questions from, we do have a question. Sorry.
I'll add one more. On the same topic and piggybacking off of what Roger was talking about the proportion. So if I'm understanding correctly, the proportion of taxonomic errors is the number of taxonomic errors as a proportion of the total number of errors.
Including like phonological and everything else, yeah.
Yeah, so just the number of errors, and this is the proportion that's taxonomic of them. So in that sense, in some of the work we've done before, that does introduce some complications because that means that taxonomic and thematic proportions are related or dependent to one another. So if somebody makes 70% taxonomic errors, by definition, they can only do 30% thematic errors, right?
Yeah.
Or the other way around.
Yeah.
I wonder if you've tried and if it gives the same results by looking at it as a proportion of number of correct items or as some sort of different proportion where they're not dependent on one another.
Yeah. Yeah, that does make sense. And it is, they would be dependent on each other. We could also view it as a strength of the proportion method where if somebody makes 9 taxonomic errors, then it kind of highlights, compared to like one thematic error, it kind of highlights that difference because you have a proportion out of the total errors. But yeah, like I kind of mentioned before, I think it will be very important for me to try this using alternative methods of actually counting the errors, right? And see if it pans out the same.
Sorry, just maybe one last then, because so piggybacking on the piggyback. What you pointed out at the end is on the distribution of the errors, right?
Mm-hmm.
So you don't have people who only make thematic errors,
Yeah.
right? If they make thematic errors, they're already also making taxonomic errors,
Yeah.
and not the other way around. So that suggests that it may also be a matter of severity.
Mm-hmm.
And if that's, which probably interacts with this in some way, right? So even if you take out general aphasia severity, how does that affect doing the kind of Friedman-Lane, analysis that you do where you tease them apart, right?
Yeah.
If you're talking about a gradient severity effect where you start with thematic errors, and if you get worse and worse, maybe even, you start making taxonomic errors, then it also wouldn't really make sense to look at these as two qualitatively different areas.
Yeah, it's true. So I think we can kind of look at this and maybe talk about your point a little bit. So like if they're really severe, does it make sense to look at them differently or in proportion? We actually see that it's almost the opposite. So people that have preserved, so this is just PPT, but people that have preserved semantic tend to make a lot of, or a high proportion of taxonomic or thematic errors. So what does this mean? So anybody who gets below like a 45 on the PPT has a pretty severe impairment just generally. So these people that have very low error proportions, semantic error proportions, that means that they're making a lot of other types of errors comparatively. So there's a lot of things going on, right, behind the scenes, and it is tricky to figure out what's the best way to measure these things, for sure.
Thank you.
Yeah.
That's it then. All right. Thank you very much, everybody. Thanks, people online. Thanks, Dr. Riccardi. And we're looking forward to seeing everybody in 2 weeks' time for Born Castle's talk.